Hello again. In this video, we're going to be looking at biological molecules with a focus on carbohydrates. So these are the main topic areas or the main sections to this topic. First, we're going to be looking at the elements that make up biological molecules. We're going to be looking at the four main groups of biological molecules. We're going to focus heavily on carbohydrates. We're going to talk about how carbohydrates are grouped. We're going to discuss what carbohydrates look like at a molecular level. We're going to talk about the different isomers of carbohydrates and what isomers are. And then we'll conclude by looking at simple sugars and how they form bonds with each other. That will be the first half of the worksheet up to page 5, and then I will cover the rest of the worksheet in a second video. Okay, so you want to get the worksheet ready and begin to fill it in as you watch the video. So, most biological molecules are made up of three main elements. They are carbon, hydrogen, and oxygen. Of course, there are other elements that may be found in different biological molecules dependent on their function. So, for instance, some amino acids and therefore some proteins contain sulfur, or they might have things like iron as functional groups that give them the ability to carry out a particular job. So the main families that we're going to be, or the main groups of biological molecules that we're going to be focusing on are these. So we'll start off with carbohydrates. And then we'll go on to lipids. That will be in a future video. And then on to proteins. And then finally, nucleic acids. All of these groups have specific functions, and we'll deal with those in separate videos. As usual, I've added a QR code at the bottom right of this page, which will take you to another helpful YouTube video taking you through some of this stuff. So now we're going on to page 2, and we're going to go on to the main bit of this lesson, which is on carbohydrates. So carbohydrates will be the first group of biological molecules we're going to be focusing on. As their name suggests, they're made up of carbon, hydrogen, and oxygen. The empirical, or the most basic formula for simple sugars, basically a formula that gives you the ratio of the different elements within the molecule, is CnH2O. So an example of that would be something like glucose, which is C6H12O6. So the ratio is 1 to 2 to 1. So for every carbon atom, you'll have 2 hydrogen atoms and 1 oxygen atom in the same ratio as the carbon atoms. There are various levels of complexity for carbohydrates. The most basic carbohydrates are simple sugars. So we're going to start off with simple sugars and talk about what they are and what their jobs are. So simple sugars are the monomers of more complex carbohydrates, basically the building blocks of more complex carbohydrates. So the three main types of simple sugar we're going to be focusing on are shown or listed below. So we'll start off with trio sugars. So these are sugars with 3 carbon atoms. They tend to be metabolic intermediates. So that's a fancy sounding term, metabolic intermediates. What does it actually mean? It means that basically these are molecules that exist for short periods of time in chemical processes on the way to becoming other molecules. So an example of a triose sugar would be something like glycerate phosphate. And we'll talk more about that in the future. And then you've got pentose sugars. As the name suggests, these are made up, or these have 5 carbons. So these are sugars with 5 carbon atoms. These form part of the phosphate or sugar phosphate backbone of nucleic acids. Don't worry about what that means for now. When we get onto nucleic acids, you'll see what I mean. There are 2 main ones you'll need to know. In fact, there are 2 that you need to know, and they are deoxyribose in DNA and ribose in RNA. And then finally, we're going to look at hexose sugars. So these are 6 carbon sugars. And they have lots of different functions. Including being building blocks of more complex molecules. So examples of these would be glucose, and galactose. So here we're going to look at two isomers of glucose. What do I mean by isomers? You might have come across the term isomers in chemistry. Isomers are molecules which have the same chemical formulae or formula, but a different structural formula from each other. So if we look here, can you spot how alpha and beta glucose differ from each other? So alpha is on the left, beta is on the right. I'll give you a second to think about it. So you've probably noticed that at carbon number 1 here, so that's carbon number 1, which I've just shown with an asterisk. In alpha glucose, the hydrogen atom is on top and the hydroxyl group is on the bottom. But in beta glucose, those flip around. So the hydroxyl group is on top and the hydrogen atom is on the bottom. So I'm going to draw alpha glucose in the space I've provided in the box there. And once I've done that, I'll give you an opportunity to draw beta glucose. So let's draw alpha glucose. So here I'm going to show all of the atoms, including the carbon atoms, in the structure. Okay, so let's start putting in all of the hydrogen and oxygen atoms on this structure. So as I said, in alpha glucose, the hydrogen atom is on the top and the hydroxyl group is at the bottom. And we number the carbon atoms in the ring, starting with the first one after the oxygen atom and then working clockwise. So 1, 2, 3, 4, 5, and 6. Okay, so you should have a go at filling in the box for beta glucose. So on to the next page. So the simple sugars that we have looked at so far are monosaccharides. What are monosaccharides? It comes from the Greek words for one, mono, and then saccharide, which is sugar. So these are single sugars. And then essence, the simplest, carbohydrates. And all other carbohydrates are made up of these. Disaccharides, as their name suggests, are made up of two monosaccharides bonded to each other. So the sugar is made up of two monosaccharides. Bonded to each other. So let's complete the table there, showing what the component monosaccharides are for each of the listed disaccharides. So maltose is made up of 2 glucose molecules. Sucrose, on the other hand, is made up of glucose and fructose. And then lactose is made up of glucose and galactose. As you can see, all of those 3 main disaccharides have a glucose molecule in them. What causes them to differ from each other is the second molecule within the structure. So at the bottom of that page, you've got a diagram showing you how maltose is formed from 2 alpha glucose molecules. You can actually form maltose from beta glucose molecules, but in this case, we're looking at forming maltose from 2 alpha glucose molecules. And as you can see, in order to get the 2 alpha glucose molecules to bond to each other, you need to lose a water molecule. And you can see that water molecule here. Okay, so you lose a water molecule. That's called condensation. And you can reverse that by a process called hydrolysis. And we'll talk about that on the next page. So, as I said, this sort of reaction is known as a condensation reaction. So it's a condensation reaction. Actually, let's put that in red to make it stand out a bit more and make it easier for you to read when you come to revision. So why is it called a condensation reaction? It's called a condensation reaction because a water molecule is lost to make the bond. Okay, so let's think about this. What are those bonds called? And if we look here, it's actually labeled in the diagram. They're called glycosidic bonds. This is important. You need to know what the name of the bonds formed in all of the major molecules we'll be looking at are. So in carbohydrates, the bonds are glycosidic bonds. And in this case, it's specifically a 1,4 glycosidic bond. As you can see, the bond is formed between the first carbon of the alpha glucose on the left and the 4th carbon of the alpha glucose on the right. Okay, so if we go down to the bottom, you can see why the structures of alpha and beta glucose are relevant. So if you look there, on the left, you can see that when you have a series of alpha glucose molecules forming a chain, they all have the same orientation. Basically, they're all the same way up. But when you have beta glucose molecules forming a chain, every other molecule has to flip over. You'll see why that's relevant when we look at polysaccharides in the next video. But for now, just pay attention to the fact that every other molecule of beta glucose in the diagram on the right is flipped over. And you can see there that, for instance, the carbon 6 group on that first alpha glucose is on top, but on the second it's on the bottom, then on the third it's on top again, and then on the fourth it's on the bottom. Okay, so the joining up of monosaccharides can be reversed. What is the name given to such a reaction? And the name is a hydrolysis reaction. Yet again, sounds a bit fancy, but actually it's just from Greek. And hydro for water, and lysis means to cleave or to split. So hydrolysis is the splitting of a molecule using water molecules. So splitting a molecule using water. So what is chemical potential energy? You've probably come across potential energy in physics. This is slightly different. So chemical potential energy. This is the energy stored within the bonds in a molecule. This energy can be released, e.g. in respiration. Okay, so how do organisms make use of chemical potential energy? As I've already said, by using a process like respiration, either aerobically or anaerobically. So by using respiration, aerobic or anaerobic. And you'd have come across respiration at GCSE. It's a chemical process used in cells to release energy from biological molecules like glucose. But that energy isn't just released freely into the cell. Instead, it's trapped within a molecule called ATP, which we'll talk about in the future as well. So the energy from respiration is trapped or stored in ATP. Okay, so that's where I would like to stop in this video. We will go on to the second half of the worksheet in the next video. So let's do a quick summary of what we've done so far. So biological molecules are mostly made up of carbon, hydrogen, and oxygen. The main groups of biological molecules we'll be looking at are carbohydrates, lipids, proteins, and nucleic acids. The most basic carbohydrates are known as simple sugars, the building blocks or monomers of more complex carbohydrates. And then we looked at the three main groups of simple sugars. We had trio sugars, which, if you remember, I said are metabolic intermediates. They exist for short periods of time on the way to becoming other molecules. And then we talked about pentose sugars, which have 5 carbons in them. The trio sugars had 3. So pentose sugars have 5 carbons in them. For example, deoxyribose in DNA and ribose in RNA. They form part of what we describe as the sugar phosphate backbone of nucleic acids. And then finally, we talked about hexose sugars, which have 6 carbons in them, and they can be used for as metabolic fuels for things like respiration, or as building blocks for more complex molecules. So an example of that would be glucose. And then we went on to talking about isomers of glucose. We focused on alpha and beta glucose. So alpha and beta glucose are isomers of glucose because they both have the same chemical formula, C6H12O6, but their atoms or constituent atoms within the molecules are arranged differently, so they have different structural formulae. Why is that important? Because it means that they have slightly different properties when they form chains. So we looked at an example of that with alpha glucose. When it forms chains, all of the alpha glucose molecules are in the same orientation, the same way up. Whereas when beta glucose forms chains, every other beta glucose molecule flips over. That's going to be very important in the next video when we talk about polysaccharides. And then finally, we looked at disaccharides of carbohydrates, which are made up of two monosaccharides bonded to each other. And we talked about the two different reactions involved there. You can have condensation reactions, which involve making a bond by losing a water molecule. And then you can break those bonds by reversing the process and adding a water molecule across the bond to bring about hydrolysis. So that is where we will be finishing off this lesson. If you found this useful, it would be helpful if you could like the or press the like button below, and you can also subscribe to get notifications of any future videos when they're added. I hope you found this useful, and please come back to watch the next video in which we'll complete that worksheet. Thanks for watching.